Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, we'll be talking about lac operon. I've made this video a couple of years earlier about the lac operon in PowerPoints, but this time I'm going to make it live. Now, we'll talk about lac operon, and we'll also talk about lac operon regulation. And we'll also talk about some other modes of lac operon. If you watched my video on prokaryotic gene expression, you know the basic thing about it. In prokaryotes, the gene expression occurs in a polycistronic fashion. You know what we mean by polycistronic? That means under one promoter, multiple genes are placed. So the expression of multiple genes are mediated by one promoter that is present in the DNA. That is very, very important for prokaryotes. While in eukaryotes, a specific gene have a specific promoter, right? It varies with number of genes. Now in this case of lac operon, we'll also have an operon contribution, like operon construction. So let me draw that construction before, like let's say this one, okay, and we have a promoter. So let's start with, this is the promoter region, P for promoter, and we have some region called operator, if I draw it or not, whatever it is. And then in the downstream of it, in the downstream of this promoter operator region, there are structural elements. Structural element, there are three more structural elements here: lac Z, lac Y, and lac A. Three different structural genes are present. On the upstream element of promoter, there is lac I and the promoter for lac I. Okay, so this is the constituent of the lac operon. Okay, the complete structure of the lac operon. Now while we are studying operons like lac operon or tryptophan operon or arabinose operon, we need to understand this first, understand how the genes are organized, because this will help you to understand the whole process. Now in this case, the idea of lac operon, let's begin with that. It's very, very simple how the lac operon works, and let's think of that without being watching the board or stuff. The idea of lac operon is breakdown of the lactose. Okay, now why we have these three genes? The ultimate goal for the prokaryotic cell is to produce three protein products from these three genes. What are those products? For example, here lac Z produces beta galactosidase enzyme, lac Y permease enzyme, lac A transacetylase enzyme. These are the three proteins that these three genes will code for. This is the idea. And why we require these three enzymes? Beta galactosidase helps in breakdown of lactose. So it helps in the lactose catabolism in the prokaryotic cell. Okay, and you know lactose is a sugar, so it can act as an energy source for the bacteria. So if you are looking at this operon model inside E. coli, we know E. coli can take lactose as a sugar and it can break down lactose and can develop energy inside. Now here, for breaking down lactose, they need beta galactosidase. Permease is a membrane channel protein which will be required to uptake lactose from the environment. Transacetylase also help in somewhat uptake of those lactose. It's the role of transacetylase is not completely clear yet, but we know it is something to deal with uptake of the lactose inside the cell. So once the lactose is inside the cell, beta galactosidase can break it down and they can use lactose and break down content of lactose to develop energy inside the cell. So this is our idea. So now let's imagine if we have lactose present. Let's say this is lactose. If there is lactose present in our body, let's say there are a lot of lactose present in our body. If that is the condition, if lactose present in our body, or let's say in bacterial cell. The lactose is present in the environment. Now bacteria wants to take energy out of it. The most important source of energy and preferred source of energy is glucose for any bacteria. But let's say the environment do not have any glucose, they have lactose present, and the bacteria want that lactose to take inside and produce energy from it via metabolism. So now what they will do, they will try to break down lactose. So they will uptake lactose and break it down to generate energy, right? Now what they have to do to take up lactose and break it down to generate energy, they have to synthesize beta galactosidase, produce permease and transacetylase. So they need these gene products to uptake lactose and break it down. So what they will do, they will turn on this operon. So if they turn on the operon, that means they keep producing these enzymes, that means they can uptake lactose and can break them down, right? Now let's imagine there is no lactose present in the environment and there is no lactose present inside the bacterial cell as well. Is it good to turn this operon on? Do you require to turn operon on if there is no lactose at all present, or it's present in very low scale? The answer is no, because in that case, if there is no lactose, there is no need to produce beta galactosidase. What beta galactosidase will do if there is no lactose? What permease will do if there is no lactose in the environment? So when there is no lactose or lactose in very low scale, they want to turn off this operon. So if lactose present, operon on; if lactose not present, operon off. It's a positive regulation created by the presence of lactose and the concentration of lactose in the medium. Okay, so let's see the whole process and how it is actually regulated. We say that we have to turn on when the lactose is there, turn off when there is no lactose. This is the thing that we understand till now. So let me write it down. So lactose, turn on; no lactose, turn off. But we don't know how the mechanism, what's the mechanism of this process. Now let's see the mechanism. You see here in this complete operon system, we have these structural genes, we have promoter and operator for that structural genes. So now you see this part, if you look at this part of the gene, 3 different structural genes expression is controlled under one promoter. That is why it is known as polycistronic expression of the gene. While there is another promoter and another gene present upstream of these structural elements. That is known as LAC-I gene and LAC-I promoter. What is the function of LAC-I gene and LAC-I promoter? LAC-I means inhibitor. So this is a lactose inhibitor gene. So if we transcribe that gene, it will produce, like if we translate it to the product, what it will give us? It will give us LAC repressor or LAC inhibitor. Okay, it will provide us LAC inhibitor. So what will happen as it is present upstream of it? So normally it is designed in a way that this operon will be turned off. Think of that. In this bacterial cell, normally this LAC operon is in default turned off. The expression the LAC operon is turned off. How? Because all the time LAC inhibitor is produced, because it is the promoter of LAC inhibitor, and this whole process is independent of LAC operon. So they keep producing LAC inhibitor, LAC-I, and LAC-I protein can go and sit someplace here in the promoter region. So as a result of that, it will not allow any RNA polymerase to sit. So there is no transcription from the DNA element here. Remember that. So let me draw it here. This is the LAC inhibitor, for example. It goes and it attaches there. So RNA polymerase don't have any access to the promoter, so there is no transcription of the structural genes. Okay, this is by default because LAC-I is transcribing all the time. Now the question is, so for turning off we don't have to bother. If there is no lactose, this whole scenario will be going on and on. Now the question is, we need to turn on it sometimes when there is lactose in the medium. So now you see, if there is lactose in the medium, in that case lactose can interact with this inhibitor molecule, and upon binding of lactose to the inhibitor, it changes the structural conformation of LAC inhibitor. As a result, it loses its capacity to attach to the promoter element. So as a result, when there is lactose, lactose will bind. Not exactly lactose, but if there is lactose present, lactose is slightly modified chemically and converted into allolactose, and that allolactose molecules can go and interact and bind with inhibitor. So what happens, let me draw the allolactose. Let's say this is the lactose. When it attaches to the inhibitor, let's say the structure is changed, inhibitor structure is changed. Normally, let's say the inhibitor structure is something like this. Upon binding with the allolactose, the structure is changed. So the active site with which they are interacting with the promoter region is now blocked after the binding of allolactose, right? So as a result of that, this inhibitor which are bound with promoter region, upon binding of allolactose, they just drop out from the promoter. So promoter is now free, okay, because allolactose wipe this inhibitor out from the promoter. So promoter is now free. RNA polymerase now can bind to the promoter, and it can transcribe the structural genes. So the operon is turned on, structural genes are transcribed, proteins will be made. So now we can utilize the lactose inside the cell. This is the idea. Naturally it is turned off, but when there is lactose, it binds with the repressor, the inhibitor, changes its structure, it no longer can bind. So promoter is free, polymerase sit, transcribe, proteins are made, and then we can use it to break down lactose. So that is kind of justified. If there is lactose, operon on; no lactose, operon off. But in default, it's turned in the off state by the inhibitor. And you know, the inhibitor molecules they are kind of synthesized constitutively in very low scale throughout the time. So this is how the LAC operon is turned on and LAC operon is turned off. In the next video, I am going to tell you the regulation of this LAC operon by the presence of other sugar molecule like glucose, because you know glucose regulates this whole process in slightly fine tuning process, and we will talk about that later. It is also known as catabolite repression. The presence of glucose will not allow LAC operon to turn on. It is known as catabolite repression. We'll talk about the role of glucose and lactose during the process of LAC operon in the next video. So if you like this video, please hit the like button, subscribe to my channel to get more videos like that, and definitely share this video with your friends. Thank you.